Yeah, I can see and then hear you. Thank you.
Yeah, thanks so much for invite, Fatih, and also from immunotyping core facility. So today I will tell you a bit about nanopore sequencing, actually just for you to get a grasp, because it's a bit of more physical method rather than pure biochemistry as with some other methodologies for sequencing. And as Fatih said, I'm now HDMI postdoctoral fellow and EMBO postdoctoral fellow in Shostak Lab here in U Chicago. So basically the outline of our talk would be the following. First, I'll tell you a bit more for you to understand what is nanopore sensing, what's nanopore sequencing, what are nanopores in general. Then we will talk about the current frontiers of nanopore sequencing, and I will give you some maybe piece of advice how you can start your own nanopore sequencing experiments as soon as possible. And here on the slide you can already see, so this is the nanopore that this company, Oxford Nanopore Technology, that's only commercial available nanopore sequencing so far, uses. And we will go actually very deep into the mechanism and how it actually works. Just to give you a brief my background, because that would affect also my claims in this presentation and my beliefs. So I did my PhD in Cambridge, actually working in the Nanopore Lab, one of the founding Nanopore Labs. And we were mainly doing nanopore sensing. You will see what does it mean, but we studied alternative DNA structures, RNA folding, RNA isoforms, protein RNA drug interactions in native molecules on the single molecule level. So for me, that turned out relatively productive. So, and I really enjoyed this nanopore journey. We also quite collaborate with Oxford Nanopore Technologies, which is the company that Fatih mentioned. And now I moved actually to Chicago, where I'm at the moment, actually working also on the nanopores, but for sequencing side. So we try to see if we do some chemistry on RNA and DNA, regardless of the type of chemistry, what does that to RNA and DNA. So we want to map these modifications using sequencing. And when you maybe heard about nanopore, you will first thing that you probably understand is a pore. So all our cells, in all our membranes that are shown here in the like plasma membrane, we have a lot of proteins that serve various transport functions. So one of them can transport ions, like ion channel. There are some that can transfer water, like aquaporin, or even like more complex ones that can transport sodium amino acids. But what we are talking about are actually the pores. And these pores actually originate from pore-forming toxins. So bacteria want to, let's say, enter the red blood cells. So it creates one of the first discovered pores that's called alpha hemolysin that actually can insert in the membrane of its host, create actually unselective pores. So now here there is no selectivity in terms of chemistry. There is only selectivity in terms of size. And I will just give you two examples of the pores. On the left side is one used for sequencing of the Oxford Nanopore Technologies, the device that we will actually talk about today. So it's basically this one here. And on the and then if you actually have this pore way larger than actually the diameter or around the diameter of single-stranded RNA or DNA, which is around 1 nanometer, then you can actually doing a sensing. You can basically see what's on DNA or RNA. So you can see bigger structures, but not see the sequence. That's why we distinguish sequence and sensing. But how do nanopores work? Actually, it's so simple. Probably if you just think about it and try reading it, it sounds like very from practical eyes. So if you take any cable, let's say like this one, and you can cut the wire in half and you apply the voltage. So you expect the current. So there's basic Ohm's law. So the point that's the thinnest would actually have the highest resistance. And that's why the current, as shown here in this cartoon, would actually be determined exclusively on these small constrictions. And how the nanopores work is kind of, here we have movement of electrons through the cable once we apply the voltage. But here we also apply the voltage, just we actually have ions. For example, kitchen salt, sodium chloride. So that sodium goes, let's say, to negative electrode and chloride goes to positive electrode. But the only way they can go is through actually this small constriction that we call nanopore or nano-size constriction, where actually we measure the current. And that's how the nanopore works. If you, for example, just apply positive and negative voltage, you only have a nanopore. I will explain you what this thing on top is. Then you will see very quick signals, like in microseconds, milliseconds. And this sequence, in that way, if you pass DNA just through the pore, we can't really see it sequence, or A, C, T, G sequence. And therefore, the company's main invention was actually slowing down the DNA or RNA using a motor protein. Motor protein is a protein that can actually translocate, as you can see here, single-stranded DNA. So we have double-stranded DNA, it unwinds it, and it pushes one strand unidirectionally through the pore in very slow manner. So now instead of microseconds or milliseconds, we actually measure current over time in like seconds. And that way we can actually really see in this small constriction, as is shown here in where my laser pointer is, we can see what letters are passing through. So basically what this method is based is single molecule. So as you can see here, there's one pore and we measure one molecule. Of course, the device parallelized, you have like array of pores, like hundreds and thousands even of pores. So we measure ionic current because ions are going through over time. So once the molecule, as you can see here in this snippet, as the DNA is going through, you see this strange pattern, like going up, down. So when something is in the pore, then it actually blocks the pore. It means like you cut the cable even more. And each of the letters or combinations of letters create a distinct signal that using some, at least in the case of Oxford Nanopore Technologies, using some machine learning algorithms, we can actually decipher from this strange pattern, like with the spikes down, up, we can decipher what's the sequence of the molecule that we are sequencing at the moment. And that's why this method is called resistive pulse sensing, because we really, what we detect is resistance, or actually what blocks the pore. And when we talk about nanopore sequencing from tires, today I will mainly focus on DNA and RNA because that's the ones that are commercially available. And also, if you're more interested in the protein sequencing, that is now also very hot topic in the area, you can also, I wrote one of the perspectives to my former supervisor. And you can read actually also that now we are moving forward trying to actually sequence proteins, modifications on proteins, modification of RNA and DNA. And some of those are already available actually within the device that also the core facility will provide you. When we move more to technicalities, so there are two types of devices or multiple types, but all of them are based on using the flow cell. So this is one of the MinION type that doesn't need computer. And then here is actually the flow cell that we can actually take out. And the only difference between these devices is actually how much flow cells you can have. So MinION, or the device that I'm holding, we can only have one cell. That means you have, you can, depending on your purpose, if you have very large genome of plant and you want to sequence it, or a lot of clinical samples, then you can use actually Promethean or Gridian. It's just fancy names for the device that does the same purpose. And then you can actually parallelize. You can do more measurement at a time. So it's kind of like similar to Illumina next generation sequencing, where you actually just run more experiments in the same time. So in that sense, you can either have like very portable device, because this device you can plug in any laptop or and go anywhere in Africa, Asia, South America, anywhere that you want on the field. Or you can actually have something more that's clinically relevant and you want to do high throughput sequencing. So the simplest way how to include, if let's say I now today want to do experiments. So basically I would need to buy a device or to use device that's available in core facility. You will need the flow cells, and you will need actually the kit for the preparation of your sample. So nanopores enable direct RNA and DNA sequencing. If you want, of course, you can do PCR, you can copy it into multiple copies, and therefore you can actually have even more high throughput. So first step is actually library preparation for RNA or DNA of any kind. And usually the kits provided by the company can guide you actually in very detailed step-by-step manner, even going to like the tips and tricks, how to avoid the introduction of bubbles in device and so on. So the protocols you can only access once you log in and create an account on the company. And the kits you can buy directly from the company regarding of the type of experiment that you want to do. For example, for RNA, you can do direct RNA sequencing, meaning actually you sequence RNA, not copy of RNA. But you can also use the one that uses cDNA or complementary DNA that's synthesized from RNA. Of course, it affects the output and throughput of your experiment. On the DNA side, they created a lot of different platforms. One of the platforms is, let's say, rapid DNA sequencing that you need actually 10 minutes to get the information if your molecule, gene variant is present in sample, and also depends on your actually goal. So there is a lot of DNA kits that are very specified to certain application, including like plasmid sequencing, or 16S ribosomal gene sequencing, and so on. So once you actually prepare your molecule, that usually takes for RNA, at least it's relatively quick. It's like 2 hours protocol with all the enzymatic reactions, adding adapters, of course, for sequencing. That's like a sequence that you need to ligate. And usually it's all provided in the kit, except these external enzymes that usually you should pre-order. And it's also written in kit what you would need. Then you need to prepare a flow cells. So flow cell is this thing that I showed you just a couple of minutes before. So it's this, let's say, tiny device. On the bottom, it has electronics. That electronics enable reading ion currents and converting it and transferring to device. And then on this side, you actually have a flow cell. And the flow cell has many compartments, basically. But here is where your sequencing is happening. So there is a platform with hundreds of pores embedded in the polymer membrane. And then you basically add your sample that has this motor protein that then sits on the pore and gets translocated or gets through the pore. And in that way, we can do sequencing. And then here, the most important port is actually this one here, where you can actually add your sample. You need to wash cell and so on. First, of course, you need to prime the cell or prepare the cell. So you need to clean it up from the storage buffer, usually some glycerol and some mineral oil. And after that, you actually just inject your library. It's very nicely guided through you in the workflow. And then once you basically put it inside, you can just actually mount on the device. And mounting on the device is relatively made, so you can't make mistake. There are 2 holes going here, so you just slide it below. You can push it inside and press, so there's a good connectivity. And that's basically everything that you need to do before. So it's relatively way faster than Illumina. You don't need to care how many times do you rotate up or down. You don't need to really pay attention, oh, what's the concentration? Do I have too much or too little DNA or RNA? So compared to Illumina, there is flexibility, because if you have a little amount of molecule, little input material, you will still have a data, but just less data, right? If you have too much material, you might not have a full ligation, but you don't care because nanopores would not translocate those molecules. So in that sense, it's really nice for you because the library preparation is really made for dummies. So you don't need to have extensive experience to know how to operate like machine. And literally you can bring that here or move within the lab and it's completely fine. Or even move from someone else's laptop to your laptop if you want this smaller device. So in that sense, this is the 2 steps that you do. once you go inside on device, you can actually start the sequencing. And when you start the sequencing, you have a certain options, of course. And some of the option is which key do you use? Is it DNA or RNA? Also, the flow cells can be different for RNA and DNA because they use a different nanopores. So all these things you need actually to pre-select before you actually even start preparing your library. You should have like correct flow cell for correct target of interest. And then you basically just follow it. You can even put even directly reference sequence. So that's a sequence that, for example, you are looking for in a bunch of transcriptome, genome of your sample. And therefore, this actually, in a sense, enables you to actually immediately see how many molecules of those that you're looking for are actually in the sample, because the sequencing compared to Illumina here is actually real time. So you don't sequence base by base, you sequence actually molecule by molecule, which is a different. So that actually enables you to actually monitor the real-time data. You can see what's going on. You can see if the length of your sequence is correct. And if you are not happy, or if you already receive your data of interest, you can literally stop the measurement and run another sample. That's simple as that. So it's very relatively quick. You can even, like in 2 hours, if you do plasmid sequencing, some people do those. It's actually very convenient. You have plasmid sequencing kit, you can pull 100 samples or 96, and basically you can just run them together. And once you get the sequence that you're looking for, you can just stop the measurement. You don't need to get so much data that you don't need. So it's actually, in that sense, very nice and efficient. I will let you after. There is one trick about the sequencing. It's called adaptive sampling. Adaptive means, okay, I'm looking for COVID-19 or SARS-CoV-2 sequence in the sample. And I just want to confirm if this patient is positive to that and what's the sequence. So you can actually tell the software, and that's automatic function actually. You can just select it in device and say, okay, this is my reference sequence. If Nanopore is sequencing something that's not my reference sequence, just push it out. So that increases your output. You get more data that you want. And it also enables that you finish your sequencing earlier than if you just sequence everything that that is inside. And we'll talk about the data analysis. By data analysis, actually, there are two parts. One that actually you can done manually if you prefer, but actually the software can do, and device actually does that in real time. So you're sequencing and you already do the so-called base calling. Because if you remember, initially, we have this nucleotide sequence. But actually, before the nucleotide sequence, we usually have these current traces, so current over time. So base calling means from this current, we actually try to extract what is the sequence of that molecule. And then you can do your data analysis, and we will actually talk a bit more about that. But just for your overview, you have people, if you present, there are these 100 possible techniques, which one we should use, why we should use. So one of the techniques that's most commonly used for sequencing today is called Illumina, or Illumina next generation sequencing. So what's actually the difference between Illumina and Oxford nanopores? So Illumina, until actually this year, actually had one extremely important feature for people who wants to do sequencing. It's higher accuracy. But actually, that's by the claim of both companies, it's not anymore holding the ground, because the last DNA sequencing nanopore actually has identical accuracy as Illumina. So therefore, actually now is the question, does Illumina becomes obsolete? It also depends a bit about your preferences. But what nanopores are good in, they actually really brought a lot of advancements in the genomics and omics in general. And it is that they can do long reads. So Illumina can read 300 base pairs in both directions. And actually, Oxford Nanopore can read long reads. What does it mean? That means that you can read the whole genome of bacteria within one go. That, of course, was amazing because it's all one of the worst problems in a genome assembly, particularly of the humans and eukaryotes in general. And that's these repeated elements. For example, ends of our chromosomes and center of our chromosomes are repeats. They're called telomeres and centromeres, respectively. And actually, until the Oxford Nanopore was introduced, we actually never sequenced fully the human genome until actually the last year. So that was the last time that we actually got the full sequence of all our DNA in the cell. And that was because of the limits of the Illumina. Because if you have like 1 megabase of repeats, it's impossible to read and to assemble because you don't know where they go. Also, one thing that I preferentially love about nanopores in general is actually that you can do native DNA and RNA sequencing. So you don't need to copy it. So if DNA has a modification, if it has a DNA damage inside, if you have RNA and it has RNA modifications, methyl 6 adenosine, and so on. So all these modifications that you actually are disease relevant, you don't lose them with Oxford nanopores. And you can actually sequence them, some of them. And even if there is no algorithm or way how to analyze the data for, let's say, you're interested in pseudouridine or something else, you can actually wait because you have a raw data, and we'll talk about that. You can save the data until someone just publish an algorithm and you can actually implement it in your pipeline. So that means that you can do epigenetic and epitranscriptome modification directly without any chemical assays. You don't need to convert to some and then do reverse transcription, do control. So you don't need actually here control for sequencing of modification that are, of course, available. The good thing that I mentioned before, it's portable. So I just brought it from the lab. I can plug it here and it should be completely fine. It enables real-time analysis. That's also very important. These two are very important for tracking epidemiology and epidemiology in general, for tracking how the infectious diseases are spreading and so on. And actually, Oxford Nanopore, this whole thing, so if you buy the MinION and the flow cells, it costs like maybe 2K. And depending on your sample, you can maybe get even like $10, $20. So people who are usually doing plasmid sequencing offsite, so you send your sample to company, actually they do Oxford Nanopore sequencing because you can get like for $10 sequence of your plasmid way faster and easier than with Illumina. So some advantages that company claims that I'm repeating myself, but just that you're aware what are the capabilities. For DNA, you can actually sequence anything from like 20 bases to 4 megabases. And particularly for short sequencing, Nanopore actually fails compared to Illumina because physically, if the molecule is going through the pore, let's see if the molecule is going through the pore and it's exiting. So would you catch the last 5 bases? They're not really that holded by this motor protein on top. So then you lose actually a bit of 5' or 3' sequence, like a couple of nucleotides, depending on your conditions. For DNA, they even develop a kit that you can do sequence of DNA like in 10 minutes. There is enrichment protocols. If you are sequencing human genome, of course, it's a massive genome. You can actually, and you're interested only in a gene related to some rare disorders. For example, I was working with my colleagues on the myotonic dystrophy type 1. And of course, you have only 2 copies of your gene in the whole cell. You don't want to sequence for 3 days. You want to sequence as fast as possible and see what's the variant of that gene. So therefore, you can actually, there's the protocols that enable you very quick sequencing. They also have PCR-free protocols to eliminate bias. So some people actually forget, but reverse transcription, polymerase chain reaction, so these 2 steps using an enzymes, introduce a bias, introduce an error. And sometimes these errors are actually too much, depending on your need. So therefore, I think it's good that you can sequence directly molecule as it is in the cell, or once you isolate it from cell. And they actually provided a lot of whole genome or targeted approach if you want to sequence for humans, C. elegans, mice, and so on. And of course, base modifications are preserved. For RNA, it's also, I think, the basically thing that you can do full-length transcript sequencing, same that you don't need a PCR or reverse transcription. You can do for methyl 6 adenine and some others, it's already integrated in device. So you can see 5-hydroxymethyl cytosine modification actually directly in the device, which is cool because you don't need to have some bioinformatic expertise, because then the device will just tell you what is inside, what's the occurrence, what's quantitatively, what's happening in your molecule of interest. You can also do cDNA sequencing. So that kit was actually discontinued, so they didn't change that on the website. So they had the option that you can sequence both RNA and cDNA of the same molecule, which of course increase accuracy. But now I think they don't have it anymore. And recently they also showed that you can do single cell sequencing combined with 10x genomics. That also I think the facility has. So if you want to do single cell sequencing, I think you can now do everything within the core. So when we talk about the data, I mentioned you before, what does your data actually mean? What you get as output? So just a reminder for you. So we apply basically current. DNA or RNA is negatively charged, so it goes through the pore towards the positively charged electrode. We have in orange here a motor protein that actually opens up DNA and basically push it inside with the help of the applied voltage. And we have a nanopore protein that actually in this constriction, so the smallest part actually reads the basis. So from that, what device collects is something that's called FASTFile file. So device will never delete raw data. That's really good because sometimes you think you might mess up your data analysis or something went wrong. You can actually go back to data and extract information from it. So FASTFile file look basically as a 2-bit file like X and Y. So you have a current intensity on the left and time on the X. And then device actually, if you want in real time, it creates this FASTQ file. This is basically just a sequence. So ACTG, CT, whatever. So that's generally what you get. So you get both the files. You can use even the FASTQ file, and then you. So we are going now to the steps here. So first is ionic current readout. So FASTFile file. So it's completely raw data. Then you have a sequence data. Then probably you will do alignment. You can use your reference. You can do de novo, and then you do final analysis. And one good thing about the Oxford Nanopore, I think they're really good in their protocols are very detailed, very nicely explained. You have even like pictures, someone how to hold your pipette, how to switch, and so on. So it's really made to the details. Of course, I think from people that I heard, I should not say more. Maybe customer service is not that responsive as for Illumina, but I think that's just their business model. Therefore, actually for bioinformatics, if you don't have any experience, don't worry, because they have this Epi2Me software. It's basically just any like PowerPoint, for example, that you just install. It's free once you have account. And then you can actually have a prepared workflow. So it tells you, oh, do you want to do plasmid sequencing? Oh, upload me FASTQ file. So you choose your FASTQ file from your sequencing results, and you just press run, and you get output, FASTA file of your plasmid. It's simple as that. So you don't really need to assemble, oh, how I should do it. Should I do this? What software should I use? Or do I have a script for it, and so on. However, for some more specific analysis, if you try to discover as we do some novel RNA modifications or DNA modifications, we might need to do that more manually. But there's a lot of resources from community that's on GitHub. So if you want to dig deeper into that, you can of course look on their website. So they actually even put all the people who create some algorithm, they actually check it and put it on website. So Fatih put in the announcement like masterful tips and tricks for successful nanopore experiment. I think that more depends, of course, on quality of your sample. So you want your RNA or DNA to be in a good quality, as it's not degraded. That RNA has a 5' phosphate, so you can do ligation and so on. So it's the same as for Illumina. And nanopore protocols are great. I really recommend actually before you actually even starting, just try to see how would your experiment would look like. And if you don't feel comfortable, then you can always talk with people around and say, "Oh, is this make sense? It sounds too simple." And they have really step-by-step guidelines, so you can track each step, how long does it take, and so on. I already mentioned briefly adaptive sampling. So it's an integrated option to search for your target of interest. So you can enter the sequence or reference sequence that you're looking for. And then if it doesn't recognize that sequence, once it's pushing the DNA through, it will just kick it out and take another one. So that saves you a lot of time, and it also gives you better data. And you can use the nanopore for a longer time. If you can finish sequencing in 1 hour instead of 16 hours like Illumina, in that sense, it's maybe better. For DNA, you can measure in parallel 96 samples, probably even more, with barcoding, at least the kit that company provides. And then actually the using of flow cell depends of the length of the time you're actually using it actively. And of course, how long you are using. Are you using for months or for days? So if you have, let's say, 100 samples, so it's a lot. It's a lot of samples. And you know that in 1 hour you get information that you're looking for. You can actually immediately just wash it and run the next sample. So if you, of course, you can just later say, oh, I want more data. But if you don't need really more data, if you're looking for very specific things, then you can actually very quickly change and put a new sample or store the cell in your fridge. There is a storage protocol, and then you can just use it tomorrow or in 1 week or 2 weeks and so on. So keep that in mind. So one thing for if you're doing direct RNA sequencing and you're looking for modifications, one piece of advice and experience, usually the throughput is lower. They are now putting a new flow cell, but it's not yet commercially available to everyone. This would actually increase the throughput, but you get less data, so you effectively need to measure longer. So basically per flow cell, you don't get like a huge amount of gigabytes of data, you will get less. So keep that in mind. Also, it is must, and one of the most important advices that is written in the protocol, and I'll repeat to you, but it's the crucial for your experiment and flow cell itself, is that you shouldn't introduce bubbles. This is like very simple electronic measurement, but if you have a bubble, then there's no current going through the pore. So you're just wasting your time and resources. And actually, in company protocol, there is one very nice trick. So you can take 1 ml pipette, you insert in this port that they call prime port, where you actually add your sample and everything. So it's actually very tiny hole here. And then you actually change your volume. So if you had, I don't know, or up change. So if you have 200 microliters, you can actually just hold your pipette and then bit increase to 230, and then the liquid from the device itself will go up. In that way, you can ensure that you didn't introduce a bubble, and then you add your sample, and then all should be good. So keep that in mind. That's probably the most important piece of advice. Just for the case of reference, I left one slide guide for newcomers. What you would need for materials: you need a nanopore sequencer, you need the flow cells, you need library preparation kits, you need optionally DNA barcoding kits, so that's some materials, and also you should look in the kits what extra tips, chemistry and reverse transcriptase, whatever you care for, you need for it. Usually it's like T4 DNA ligase and so on. So you can actually check that. So that's what you need to get before you even start. Once you start, you should read actually the library preparation kits protocol. that would enable you to use your purified DNA or RNA with specific amounts. Also, they tell you what they suggest, but you can have less or more. You follow the instructions, and then important thing is that the flow cells compared to Illumina, but probably in Illumina as well, but here is more important, can differ because there is array where maximum is like 2000 pores. But because of they're in solution, right? So you go to transport and so on. It could be from 900 to 1600. What does it mean for you? It means you will get less or more data. More pores usually means that you can get more data in the same amount of time. And the good thing about the company, they usually say if this number is below the certain pores, once you run your flow cells, you should check on device, there's bottom press flow cell check. And it will tell you, okay, it doesn't meet your requirements. It's too low. You still can sequence. Don't get me wrong. You can sequence, particularly for plasmid sequencing or something that's like low throughput need. You can actually use those, but you can then ask company to send you new flow cells because those didn't meet their criteria. So in that sense, they're really nice. So if for some reason the flow cells have lower, you still can use them. You don't need to return them. But you will get a completely new batch of those that are with a lower quality. Then you need to prime or prepare your flow cell. So usually that's just washing the flow cell with some buffer before you add your sample. And once you prepare the cell, you have your library. So you have your molecule, you have adapter with the motor protein that's actually going to go to the pore and push your sequence inside. Then you can actually go to sequencing. So you pipette your library, start sequencing run, tell what you want to sequence, what kit you're using. It's very user-friendly in that sense. And then after that, you can wash your cell to load another sample. As I tell you, you can immediately put another sample once you're happy, or store it for the next experiment. So it's, of course, up to you. And usually I think the company says 5 times, but if you're really stingy, some people actually for plasmid sequencing, like core facilities to do plasmid sequencing, they actually wait for samples together, like hundreds of samples. Then they sequence for 1 hour, then clean it, and then they use it like a lot of times until actually they get, the last pore dead. Usually we throw it way earlier, like when you reach like 100 pores, you will need to measure like a long time to get something. But for these facilities, they really know how to maximize that. And then you do data analysis. So first step is, of course, base calling, sequence alignment, and then you can do actually well-established analysis with Epi2Me. There's this free, available, user-friendly, no coding experience platform. And then they also offer some more like advanced algorithms, but for that you would need to know how to use a GitHub, how to use that algorithm, how to run it in Python or wherever. And with that, I will finish basically this first part. So then after, you can ask any questions or any uncertainties about probably facility that Fatih can answer, or regarding actually the sequencing or nanopores itself. And just if anyone is interested to doing like a rotation project or intern project or research project in our lab, so we have some projects related to RNAs in general and RNA with the nanopores and RNAs that can be nanopores. So you are free to drop me an email. It's same as my first and last name, and then just at uchicago.net. That's it from my side.
Thank you very much, Filip, for the excellent explanations about the Nanopore. Yeah, if you have any questions for the users, if you have any questions, you can unmute yourself and then ask these questions. If there's no question, I have a question. Actually, a couple of questions. For example, for Illumina, also the low complexity or low base diversity of the sample is very important. For example, if the sample has low diversity, then we need to spike in 5x. What about the nanopore?
Oh, that's very good point. I actually forgot to mention that. So keep in mind that for nanopore, you don't need to do that stuff. So let's say you don't need to have GC rich sequence. You don't need to have like balanced ATCGs. Even the last advancement or the last nanopore that company made, so it actually sequences your molecule within the pore 2 times. So there are 2 constrictions like this. So molecule goes through and it sequences here and then it sequences here. So they actually show that they can do also homopolymer sequencing. That's one of the problems with Illumina as well, where you have AAA, CCC, GGG, TTT. So in that sense, you don't need that.
Okay. The next one is also you mentioned about the reusability of the flow cell, right? We can reuse the flow cell a couple of times.
Yes.
Then the next question is, what about the design of the experiment? For example, if we have, let's say we have a multiplex PCR, and then we amplify the 16S RNA, and then we have lots of samples, right? Human samples or kind of samples. So what about the designing of sample size? What are the criteria to design the size of the sample? How many samples we can use for, especially a minion flow cell or protein flow cell?
So for the multiplexing, I can tell you for RNA, if you want to do direct RNA sequencing, multiplexing is not available as a part of the device, but you can do it if you know how to do it manually. For the DNA multiplexing, let's say 16S, let's say you're doing this metagenomic study, right? So you have a lot of random 16S sequences and you want just to multiplex. There is 2 ways to multiplex. You can multiplex different samples or you can even multiplex different molecules, okay? So you can put a barcode on very specific, like 16S from Acinetobacter baumannii, but in other case, you can say, I isolated this from patient 1 that has 100 possible pathogens inside, all kinds of stuff. And then you can actually do this multiplex barcoding. So, and then to run. In terms of the amount or how big, how you should design your experiment, that very depends. So usually the company gives you what's the minimal input DNA amount that you need to have. And it also depends if you have a complex sample, like patient sample, something isolated from the group of organisms and something like that, then you will probably have to titrate your sample. The nanopore, I think people use nanopore for a lot of stuff like that. So you can see in the most of papers very nice. So they just say, oh, we added 500 nanograms of DNA. We use the same sample. So you basically just repeat the same. It should work. Even though if you have a bit variation, it still should work. It's not like with Illumina. In Illumina, you need to have a certain range of DNA concentration, right? With nanopores, it's a bit more flexible.
Okay. Also, there are 2 questions on the chat. Can you see the questions on the chat?
Yes. So there is one question from Ruvid, if I pronounce properly.
Yes. Okay.
Any tips for keeping more pores active through the run? I'm losing a lot of pores, about 50% in just a few hours. What might culprits before this? This is relatively a simple thing. One, it's expected for you to lose the pores over the run, or active pores, as you properly said. One thing that you can do to keep them alive is actually that your sample is properly purified. So the most of the problems arise when you have too short DNAs, because they actually occupy the pore and actually kind of destroy them or use them. So one good thing is that you actually have a good initial sample. If you don't care about the short DNA sequencing, but you more do, I'm not sure what exactly you do, that might also help with the answer, that you actually just purify your DNA a bit more. And in that sense, you should get more higher pore activity. Also, one thing that why you can have a low activity is if you have too low concentration of DNA, or you have ultra high concentration of DNA, because also DNA that's not labeled with the actually this adapter sequence, would actually just go through the pore, you know? So in that sense, it might actually be destroy some pores. So you want actually that all of your molecules that you care about are properly labeled with adapter so that you follow their ratios for the enzyme towards the concentration of sample. And in other sense that your sample is clean. So if you did ethanol precipitation or something like more dirty methods, then that tends to affect, if there's ethanol, you will maybe solubilize proteins a bit or something might affect the biochemistry of the flow cell itself. So this is two things that to keep in mind, like quality of your sample and that you follow the protocol in terms of concentrations. I hope that answered the question. If not, just write a follow-up or join. So Benjamin has a question. Can you elaborate on how to implement adaptive sampling? Does Oxford Nanopore provide software that makes it easy to do? Any special computer needed? No. So it's actually when you enter device, you start sequencing. It will ask you which kit are you using. Do you have a reference sample? Can you choose it? Can you let me know how long I should sequence? You can either say how long or how much data I should sequence. So you can say if you collect 1 gigabyte, stop the measurement. That's usually for your sample enough. Or you say, okay, I only care about ultra long DNA. Let's say you care about 1 megabase. You can say until you reach this amount of 1 megabase molecules. In terms of adaptive sampling, there's a tick box that you just press with your finger, and actually it will use your reference sequence for adaptive sampling. There are some new advancements in that are not integrated in device itself, but you don't need any special computer. So it's done before you start your sequencing. You choose actually adaptive sampling as an option. You don't do it after. It's not data analysis process. So Silarine, there is a question. Is there any Nanopore specific library preparation kit for single cell library preparation, or can we use 10x genomic chromium library preparation kit to run in Nanopore sequencer? So there's the first question. That is relatively new thing. So I think it was done this year with collaboration of Oxford Nanopore Technologies and 10x. So if you just go to their website and just write single cell, there is actually workflow. You don't use any special kit. I think usually the 10x should be enough, but don't hold me fully. I haven't checked that. So I think it's usually you just follow 10x protocol, and then after that you just do ligation of the adapter, and then you just do sequencing. So it should be very straightforward. You behave as any other sample. Can we use Epi2Me for single cell data analysis? Oof. So you can do for gene expression data, but not for single cell. I think you would need to actually do as you would do for any other single cell, let's say transcriptome data analysis, if we're talking about 10x poly A enrichment from single cells. So you will need to do that, not in Epi2Me. So Rubit has a follow-up question. This was with the control lambda DNA test, and I still had a lot of inactive pores. I don't think purity should really be an issue. Do you think this is maybe a library prep issue in that case? It could be. If you see something that's extraordinarily strange, that you lose a lot of pores with the control sample, I would just actually recommend to write to the company and say, oh, this is my data. It says that most of pores are dying. Is this normal? Can you show me your data for control experiments? And then if you see the data like 10 times better, they don't lose any pores, then you can actually ask, maybe they send you either kit is too old or you didn't do proper ligation, or one of the biggest cause of the errors is actually bubbles. So that you didn't pipette well the library in the device. So you have to be extremely careful that the priming of the cell is good. But you can, if you get the data, don't run another experiment, don't waste your resources, just write to them and ask, send the data that you got. You said, okay, after 1 hour, I lost 50% of active pores. What might be reason? I ran the control sample using this kit. And they usually will tell you what are the source of these errors. But it really can depend.
Okay, thank you very much, Philip.
So you can also see Zenrun had from their experience that they have a short shelf life. Also, I think you can't measure them for 5 days continuously. Okay, so one full cell doesn't have that that large lifetime. Although, if you have very long DNA molecules, actually they have a very, because DNA molecule anchors the pores there. So it very depends on your sample. But yes, don't expect that you had in 1 day 100 pores left. So usually we go from, let's say for RNA sequencing, at least from 1600 to like 900 after like 16 hours of measurement. So that's a normal. But between 1 hour should not be that bad. Okay, so John Stelly has a question about adaptive sequencing or adaptive sampling, actually. Can you select against in addition to selecting for sequences? In selecting for sequencing, can you only select for one or many sequences, and if so, how many? Can you also select for sequences at the 5' end of the sequence? Oh, these are really good questions. So can you select against? I think don't. I think not. That's what device does. So if you select for sequence A and it reads sequence B, it will kick out the sequence B in that sense. Can you select for multiple sequencing? I think yes, but that's not that straightforward. You will need to look up. I haven't done that before, but I think it probably can be coded to do so. Can you also select for a sequence at the 5' end of sequence? Assuming that you're talking about the DNA, because DNA and RNA sequencing goes in a different directionality. So RNA sequencing is from 3' to 5' just because motor protein is different. And then DNA is 5' to 3'. So therefore you would lose, when we say 5' end, probably the last 10 nucleotides would not hold the water, or even maybe 15. So they have this 5' truncation, but you can definitely pick like a shorter sequence. You can take 20 nucleotides or 30 that you know that will be in all your samples of interest, and let's say others are just isoforms or variants, and then by that you can actually probably do adaptive sampling, but you need to give it a try.
Yeah, okay. I think so we came to the end of the webinar, Philip. There's no more question, I think.
Yeah,
There's one?
No, I think
okay,
that's,
yeah.
you responded
The
there.
follow-up question from Ro, I think, yeah.
Yeah, if I didn't answer anything, just write in the chat or just raise your hand and just turn on your.
Okay, thank you all for your engaging participation and insightful questions during this webinar. We truly appreciate your active involvement and hope to see you in our future events. Thank you very much, Philip.
Thank you, and thanks everyone for
Bye.
joining.
Thank you.